Yeah.
Are you both speaking?
Just you, but it's not, isn't it? And then we have to stop it later on.
Yeah. So that's for coming into our intrepid building. And we have Matt Brown, who's the Chief Scientific Officer of Genomics England. He's going to give a talk, and Matt and Amanda are around till 3 o'clock, I think. I'm going to have and if people are interested in meeting and discussing with them. So I think, yeah, you want. Yeah. Thanks very much.
Cool. All right. Well, look, thank you very much for coming along. It's lovely to see so many familiar faces. And I feel that there's a very strong antipathy and connection between Bristol and Brisbane particularly.
Yeah.
And it's always lovely to catch up with people again after so many years. I was just saying to George that this is the first visit I had to this unit was coming up 30 years ago. So there are probably people in the audience or online who actually weren't born at that point. How genetics has moved on since then, thank goodness for that. Okay, so what I'm going to talk about today is about Genomics England's programs in rare diseases or heritable diseases, and try and convince you that actually it's a cohort that's actually much more useful than that, and that we're actually quite a useful common disease resource as well. And then also talk with you a bit about our programs in lifting diagnostic rates for rare disease patients, and finally about what our programs are for the next 4 or 5 years, which are progressively being announced as the government announces the results of the spending review. So first up, a little bit of history. Genomics England is now about 12 years old. We were kicked off in 2013 with the government's announcement that they were going to fund the 100,000 Genomes project. At that point, I was in Australia. I don't think Amy had quite yet joined our lab, but we were sitting there saying 100,000 genomes. To quote a line from the famous Australian comedy, The Castle, we thought they had to be joking. Anyway, so nonetheless, 100,000 genomes were actually completed. Those of you who are eagle-eyed amongst you will see that that does not add up to 100,000 people, but that's because the cancer patients had a 2 times normal whole genome of their cancer plus a normal genome. So these people are counted 3 times. Whatever, it's predominantly a rare disease cohort. The patients were recruited out of the NHS. So these patients were referred in because they were considered to have a rare disease, not because they were actually known to have a monogenic disease. In fact, some of them had actually been previously sequenced and people knew that, but majority were actually just referred because they had severe disease, familial disease, or early onset disease, which are basically proxies for people having a monogenic disease. So out of the 100,000 Genomes program, that led to the development of the Genomic Medical Service, which was launched in 2017-18. And there's been a very close relationship between the Genomic Medical Service and Genomics England ever since that, such that Genomics England provides the whole genome sequencing for rare diseases and cancer for the NHS. And sequencing is performed at a centralised facility currently at the Illumina lab in Cambridge. The data from that sequencing comes through Genomics England's pipelines and basically gets all of its processing and variant calling and annotation performed by Genomics England, and then that gets passed back to the NHS, which is then used for genomic laboratory hubs to produce the reports that actually get used by the NHS. All of the patients who participate or who have NHS whole genome sequencing are then asked to consent for their data to go into the National Genomics Research Library, and over 90% of patients do. And as a consequence, the National Genomics Research Library is growing fast. We're sequencing in the region of 4,000 to 5,000 whole genomes a month, predominantly rare diseases, so about 80% rare disease and the rest being cancer. And that data is then available for researchers around the world at no cost to use for all sorts of genomics research. And there are benefits then back to the NHS through, for example, as I'll show you, a lot of diagnostic to discovery work where essentially we crowdsource the world's academic community to look at the data, come up with diagnoses, which we then verify and then pass back to the NHS to actually return to patients. And that's resulted in several thousand diagnoses being returned to the NHS. The vast majority of which, around 90% of which actually ultimately are considered accurate diagnoses and, of course, of huge benefit to the patients involved. So the consequence of that is that basically the cohort is continuing to grow and continuing to enrich itself, particularly with rare disease, but also with cancer data. But in addition to that, we're doing all sorts of other cohort studies and...
Can I click to get rid of that? Can you click this up a bit?
Hey. Done. Yeah. The cohort is growing consequently and also changing in its nature from being entirely rare disease and cancer to being one where there's a progressively increasing amount of general population data. So with the current build of the National Genomics Research Library dataset, there's about 165,000 whole genome equivalents in it, which is predominantly rare disease, but also includes, for example, a 25,000 cohort of patients who were recruited with severe COVID infection. And then we know that from our funded recruitment and sequencing program that it's going to grow over the next 3 to 4 years to exceed 500,000. We actually think it'll probably get to around 700,000 by 2030, but conservatively it's going to go over 500,000 by that point. And that's going to include growth in rare diseases, growth in cancer, but particularly growth through the completion of the generation study that Amanda's heavily involved in, which is our sequencing of 100,000 newborns, which should complete recruitment by 2027 at its current rate. So this number will probably be exceeded because we're expecting that generation study will roll on into something else with West Street announcing the intention that this actually become a general thing for English babies anyway, that this become available. And in addition to that, there's another cohort which I'll talk about towards the end called the Adult Population Genomics Program, which is a study of 150,000 adults over 50 years of age, and that has been done for preemptive pharmacogenomics, which will enter into the database. And the exact numbers that will have completed by 2030 for that are not clear, but a conservative figure is 40,000. So we think it's going to exceed 500,000, probably be more likely 700,000 by that point. In addition to that, the data types that are actually becoming available are rapidly expanding, such that it also now includes on the cancer patients already over 100,000 cancer digital histopathology images from the tumour slices of the patients, from the patients through the samples that were taken for the whole genome sequencing. It's got a large number of long-read samples, or Oxford Nanopore samples, with both genetic and epigenetic data in it from predominantly rare disease patients. And I'll talk a little bit more about this multi-omics cohort from rare disease patients, which is going to reach probably 10,000 by the end of this financial year. All right, now a unique feature of this data set is that it is whole genome equivalent. So all of the data that's in it is whole genomes, not exomes or patterns. And so for rare diseases globally, that's quite a unique thing. That results in us being able to identify a whole stack of things that have been missed to date. Nearly all rare disease genes that have been identified to date are actually in coding regions. And the extent to which that's actually because that is the major cause of rare diseases versus that's because we only have had whole exome or exome targeted sequencing available is not really clear. And people are really only now starting to get into looking at non-coding regions of the genome for rare disease mutations. So this is a great example of the potential of our data set to discover new things, which comes from two groups, one of which is Bristol-based, particularly Andrew Mumford, and the other one of which is predominantly Oxford-based, driven by Nikki Whiffen, who in parallel identified a monogenic cause for neurodevelopmental delay involving a gene called the RNU42 gene, which is part of RNA splicing mechanisms. And it turns out that this is actually either the most common or close to the most common cause of neurodevelopmental delay worldwide. So I'm presuming you must have heard of this because it's such an extraordinary story. That basically had been hitherto unrecognised by genetics or by the clinical genetics community, despite the fact that it had characteristic facial appearance. Generally, I think clinical geneticists are outstanding at pattern recognition, but this is one that slipped by. And I don't put this up to be critical of clinical genetics, but just to say, if this is missed, there are probably quite a few of others that have been missed too. And so basically, it shows that there's a lot of unmet diagnoses or undiagnosed conditions that are sitting out there, despite the fact that we've had, really high quality researchers looking at this using other methods for centuries, really. Okay. All right. So I think the change in nature of the 100,000 genomes or the National Genomics Research Library is particularly relevant because of the fact that the nature of genomics research is changing quite a bit. So I think we're seeing that researchers are generally, when I kicked off in research, mostly you used research data for your own experiments that you generated yourself or that your lab had generated. Whereas nowadays, actually, it's the converse. The vast majority of researchers are using data which has been generated elsewhere. And the diversity of research has also substantially increased. So again, back to John and I's research in the 1990s, it was largely disease association work trying to identify new disease-associated or causative variants. Whereas nowadays, I think that wave of discovery, although it's not over, has actually become less important. And the shift has been much more towards functional genomics and target identification for therapeutics. There's been an increased focus on multi-omics, and you can list a whole long list of omics there, which is partly because of the fact that we actually now have the tools that we can do high-throughput multi-omics with robust, reproducible results, but partly also because people understand that actually there's a lot more to actually working out what's going on in the disease rather than just finding a disease genetic association. I think the type of people who are using genomics data is also changing, such that, again, back in the day, people who were working in genomics generally were good at coding and could do their own analyses, whereas nowadays, actually, it's becoming much more a routine tool of the functional genomics community, and molecular biologists who are non-coders are using the data a lot more. And therefore, organisations such as ours who have large-scale genomics data need to cater for these sorts of usage. And the last thing I'm going to talk about is that, and this is a particular relevance to the change in the HRO, is that common disease research has shifted increasingly from the identification of common variants of small effect towards identification and study of rare disease-associated variants. And that this is being enabled by the fact that there are increasing data sets that are actually derived from sequencing data rather than SNP microarray data, where SNP microarray data was basically highly inaccurate for variants of less than 1% minor allele frequency. And by imputation, and therefore really were not suitable for finding these rare disease-associated variants. Whereas the shift towards sequencing-based data sets means that finally we can. So the National Genomics Research Library is a great example of the fact that actually many people with rare diseases are actually just people with common diseases, but who have a severe end of the spectrum of common diseases. So when we look at the data set, this is the pilot study from the NGERA. And on the y-axis of this figure is the diagnostic yield, the proportion of patients that we've actually returned a genetic diagnosis to. And in that original analysis, about 25% of the patients who were referred in actually turned out to have defined monogenic disease. The people who the remaining 75% or so probably include a large number of people who actually have a rare disease that we've not yet actually just identified the genetic variant, a group of people who've got environmental causes for disease, but also clearly people who have other genetic models for disease, be it oligogenic or polygenic, but which through our current pipelines, we don't ever pick them up. But we know from polygenic risk score studies that actually they're weighted towards including an excess of common disease cases. So this will be very familiar to many of you in the audience. This is from the most recent height genome-wide association study with X many million. I can't remember exactly how many million people, but basically it's at least a phenotype where gene discovery has been saturated and pretty much all common variants for the disease, for the trait, have been identified. So it's a really good way of looking at the relationship between variant frequency and variant effect and looking at the genetic architecture of disease. And so you see that in this figure, plotting one allele frequency versus effect size in bone density, that, this bone density, not height, that basically the effect size of the variants that have been identified increases as the variant frequency reduces. And if you look at the genes that are down this end of the figure, it actually includes many genes which are essentially causes of monogenic variation in bone mass, so osteogenesis imperfecta or high bone mass genes like LRP5, SOST, and WNT. And these are being picked up in other studies through whole genome sequencing and exome sequencing. These variants here, which are much more common, have been picked up purely through SNP microarray. And when you look at this by whole genome sequencing, you can pick up the whole lot, which means that now these people who are being referred in through studies of just normal population variation in bone density, you using a whole genome sequencing, you're able to basically, in the one study, pick up both the rare and the common variants, and that what was divided up based into the severe clinical phenotype before of osteogenesis imperfecta, high bone mass, versus just common and garden osteoporosis is actually now just one continuum. And I think that this case, you can see this when you look at height as well, and that it's probably the case through most common diseases that this sort of breakdown or division between rare and common diseases is artificial, and that once we move to sequencing-based gene mapping, that we will look at, that we will do away with that. All right, now I'm going to talk about work that we've done bringing together harmonizing data across major databases worldwide. So this is not, of course, all global databases, but it is a group of databases where the PIs have agreed to standard fee code definitions of ICD categories, so disease categorization, and then implemented basically annotating their data sets with fee codes similarly, using the same definitions for the fee codes so that people can work between them. And I'm going to put up some examples here to compare the National Genomics Research Library with the UK Biobank, not because we're competitive with UK Biobank in any way. In fact, we collaborate very closely with them, but simply because I think everybody in the room probably knows of the UK Biobank and uses a lot of you would use it for all sorts of studies in common diseases, including that dreadful method, Mendelian randomization, which has been the absolute bane of all journals worldwide. Thank you very much, George. Anyway, so this is, after this annotation exercise, we looked at what was the gain if you combined or if you compared the NGRL with the UK Biobank, and if you combined the two for analyses. So we identified just over 2,000 fee codes with 100 cases in Genomics England in the NGRL or the UK Biobank. And there were 209 where there were more cases in the UK Biobank than there were in GEL. And there were 325 where there were more cases in Genomics England's data bank than the UK Biobank. So for fee codes, we actually have more fee code coverage and for more diseases than UK Biobank does alone. Then if you look at the gain that you get from adding the two data sets together, because we would have expected if the distribution of fee codes had been similar in Genomics England and the UK Biobank, that there'd be a 23% increase in the number of fee codes per, or the sample size rather, in the combined data set compared with the UK Biobank data set alone. But across all of these different categories, actually the median increase was much higher than that. And the reason for that is because the Genomics England data set is so enriched for patients with disease compared with UK Biobank, which is a very healthy population data set. And this includes not just rare diseases, but a whole bunch of common conditions. So I'm looking here, for example, Crohn's disease, obesity, type 1 diabetes, anemia, insomnia, depression, pregnancy-related conditions and things like that. Actually, by adding the two, you gain way more than just the additional sample size from the current release of Genomics England's data set. Okay, so then some people suggest that this is just because we're weighted towards kids with severe young disease, but actually, irrespective of date of birth, across age ranges, there's an increase in P codes in the cases from Genomics England's data set compared with UK Biobank. All right, so then looking at some specific diseases. So for some diseases like polycystic kidney disease, most of those had actually already been diagnosed as rare diseases, but we still pick up an additional 350 or so by the fee code definitions. Whereas for other diseases like pulmonary fibrosis, which is actually predominantly a common disease with only some rare disease causes of it, we only had 175 people referred in because they had a pulmonary fibrosis as part of a rare disease. And we picked up nearly 1,000 more through the fee code definition. And so we've then been doing work, doing gene mapping with that. And this is GWAS using the NGRL data set, looking at interstitial pulmonary fibrosis, which is, a not rare and severe disease. And through our own data set, which, clearly wasn't recruited specifically for that, we identified 2 new common variants, replicated a rare variant, and identified a completely new rare variant, which we've subsequently replicated in other cohorts. So I put this up just to say, if you're working on common diseases, you should still consider using the NGRL. At the moment, at 165,000 whole genomes, it already adds significantly over and above UK Biobank for many common diseases. By the time it hits 500,000 genomes before 2030, it's going to be a real powerhouse for common diseases as well. All right, now what about diagnostic discovery? So here we've had this huge internal and external effort in trying to increase diagnostic discovery. It's been led by Dr. Susie Walker at Genomics England, and it's added now over 5,000 new diagnoses to the 100,000 genomes cohort that have been returned to the National Genomics to the National Health Service. That brought us up to around about 30% of patients from 100,000 genomes actually having achieved the diagnosis. So it lifted the diagnostic rate by about 5%. So 6 years international effort, really diligent, 5% increase. So we've been looking at the use of other technologies to increase the diagnostic rate, particularly we have funded through a collaboration with industry involving these technology partners, a program where 7,500 of our rare disease programs have had a long-read sequence, so both long-read and epigenetic profiling. They've had their a peripheral blood whole transcriptome. They've had a 5,000 proteome study done using the Illumina protein prep assay, and they are having a comprehensive metabolomic profile done by Metabology. This data will all be in the NGRL from basically Q2 next year. A lot of it is already in the NGRL, including all of the transcriptome data and 5,000 of the long reads. And so the goal of this is to see whether we can increase diagnostic rates, but also to create a functional genomics data set, and one that you will, shock horror, also be able to use for Mendelian randomization. Sorry, George. Anyway, this is just an example of the gain from long-read sequencing. This is a patient who has a monogenic form of obesity, Prader-Willi syndrome, and you can see the person has, in the single strands, you can see both the coding variation and the red bits, which are the epigenetic change, the methylation change on CPG lines. And so it's really nice because you can see here they've got a point mutation, and on the same strand, they've got basically a reduction in CpG methylation compared with the other strand, which carries the wild type and has the normal methylation pattern. And that's important because this is an imprinted disorder. So in the one section, in a strand-specific nature, you can see the coding and epigenetic variation. And that's, I think, going to be of great value in looking at functional genomics and in working out how variants actually ultimately end up causing disease. So just briefly about the proteomics study, this is completed now 7,500 patients from our rare disease programs. Another 3,000 are going to be done before the end of this financial year. And I'm just going to show you some data from the pilot group of this. So this is only 500 patients looking using just 1,500 proteins. And what we showed was that using the limited protein prep assay, that it performed as well as the somatologic and only assays would, with good low coefficient of variation and high sensitivity. We then did some really interesting fun things to see whether what we were picking up was actually real or not, noting that these samples, that the serum samples had been obtained in less than ideal circumstances. So they were recruited as part of 100,000 genomes, a study. Samples were taken and processed in sometimes academic labs, sometimes NHS labs, not using standard protocols, different collection tubes. They've been free-stored, all sorts of bad things have happened to them. So were we still able to detect things? And the answer to that is absolutely yes. So we developed a classifier for gender using the proteome levels. And you'll obviously understand what an ROC curve is. So this classifier was pretty good, but not perfect. It had an area under the curve of 0.986. And then we thought, okay, well, what about also looking at, can we predict age? And so this is a plot of the actual age versus the predicted age from the proteome of the patients. And the blue dots represent the individuals where the gender was not correctly assigned using the classification over here. And you can see that the vast majority of them are actually down in this bottom end of the graph, which are the pre-adolescents. So presumably the reason that we're not able to actually assign gender is because these people have not yet seen the effect of sex steroids through adolescence on their proteome, and that explains the misclassification. There are a few others out here which have some, where there's still gender misclassification, but it's a small fraction of the total. And we're looking at these, but it looks like these people are largely people who have body mass index extremes, and that may actually explain why their proteomes are different. So it's probably biological and not related to any issue with the assay. We then compared patients who belong to a particular disease category, such as, for example, renal disease or neurological diseases and so on, and compared their proteome with the proteome of those in the cohort that didn't have that. So this is an underpowered study because in each category there are only about 50 people. So it's 50 people versus 450 people. So really underpowered. But nonetheless, we saw experiment-wise significant difference of abundance with a whole bunch of different disease categories, such as renal disorders, vascular disease, metabolic diseases, and cardiomyopathies. Whereas in other disorders, like, for example, growth retardation, we didn't see very much at all. And when we looked at the, this is a volcano plot, and when you look at the actual proteins that were differentially abundant, they are highly relevant to cardiomyopathy, to the disease is concerned. So this is hypertrophic cardiomyopathy, the most differentially abundant protein. Mutations of that are a known cause of cardiomyopathy. Renin, highly differentially abundant, known to be a biomarker of heart failure. This protein, there's a mouse model of a knockout mouse model that gets cardiomyopathy. And down here, mutations of this gene also cause cardiomyopathy. And a particularly interesting one, this one is a protein that's reduced in patients with cardiomyopathy. And there are now inhibitors of this protein, which are a licensed treatment for a form of cardiomyopathy, suggesting that this is going to be actually useful for disease discovery, treatment target discovery work as well. So Julia Carrasco-Zanini and Claudia Langenberg then took another part of this project and compared the data from our cases with data from historic controls that have been, where there'd been bridging samples run between the two data sets. And the red diamonds here represent individual cases, and the box plots here represent the control data set. And so these cases here are all cases where they had a protein level which was lower than basically the entire control data set. I'm just going to take one example here just to show how this works out. So these two people here are unrelated people who had a reduction in a protein called setmar. And when we looked at those two people's genetic data, they had an identical rare missense mutation in setmar. And proving that a genetic mutation like this is actually the cause of disease is extremely hard to do, but it turns out they both had a very similar phenotype with a metabolic muscle disorder and rhabdomyolysis. It turns out setmar is highly expressed in skeletal muscles relevant to that. And the kicker is that there's a setmar transgenic mouse which gets rhabdomyolysis. So this is an example of how you can take what was a research cohort study and take it down to individual level and actually achieve diagnoses. So out of this one study, they lifted in that cohort the diagnostic rate by 7.5%. If that translates across the whole cohort, that will mean that we'll get 7.5% increase in diagnostic uplift versus 5% so far of 6 years of multiple people's effort to re-diagnosis looking at the DNA. We've got similar data for transcriptomes, so we think we're going to be able to get a 5 to 10% uplift in diagnostic rates through transcriptomes. And this might mean that we actually get to an actual rare disease cohort diagnostic rate of 40 to 50%. And I think we're probably going to plateau out at that point. The rest of them are going to turn out to be other things. Right. I'm going to run out of time. So I'm going to skip through this bit. I'm going to just talk about where we're going next. So for those of you who read through the NHS 10-year plan and the life sciences sector plan, you'll have seen that the government stated in that their expectation is that by 2035, half of all medical consultations will involve a genetics component. Now, of course, that's an uncertain statistic. It's a guesstimate, but basically that's their expectation of the trajectory. And if that's the case, that's going to result in a need for us to be able to run tests and provide results back and build into clinical services millions of genetics tests per year rather than the hundreds of thousands that we're talking about, which is a huge ask. And before you do that, you really need the evidence about how that's actually going to be beneficial. I've mentioned already the generation study, and that's one piece of research going into trying to assess the utility of sequencing to identify treatable genetic disorders amongst newborns. But there's clearly a lot of potential for using genetics in other phases of life. And so therefore at Genomics England, we looked at what we do next. And so what we're going to do is what's called the Adult Population Genomics Project, which is a program which we're going to perform together with the NHS and the NHS Network of Excellence based at Manchester, where we're going to study 150,000 older people. Older means over 50, which there are a few of us in the room anyway that achieve that one. In this, they'll be recruited through community level or through general practices, and they're going to have whole genome sequencing performed, and that data then be interrogated for a few different purposes, but the primary purpose is to look at the utility of preemptive pharmacogenomics. So genomics before somebody's actually prescribed your medication to guide the physician on whether that's the right medication at the right dose for the right person. And the idea here is that we should be able to result in fewer adverse drug reactions. Adverse drug reactions cost NHS England over £2 billion a year, and they're responsible for 1 in 15 casualty admissions. We also hope and are going to be looking at whether we can improve health outcomes by, for example, reducing tertiary referrals for conditions that are relevant to pharmacogenomic variation, such as, for example, psychiatric disorders. And so that's the primary purpose of it. The secondary purpose is that we will also be returning data on cancer genetic risk, so monogenic cancer genetic risk, in particular things like Lynch syndrome, for which there is currently cascade testing in the NHS, but no population level screening. And then lastly, we'll also be returning polygenic risk scores. So exactly which polygenic risk scores, we're not sure. We'll probably be starting with cancer first, but it's the beginning of basically polygenic risk scores, I think, becoming a routine part of healthcare in the NHS. All right. The last thing to say is that I've talked a lot about National Genomics Research Library. As I said, it's free of charge to academic users, including currently the compute. It's really easy to sign up to join it. We've simplified the process so that what used to be what was called the GSIPs, of which there were 38 of them, we now have 8 different communities. These communities are really active and are holding all sorts of events, from conferences to online education programs and so on, collaborative networking efforts, and I've broken those down here. And if you want to get access to it, you have to join the research network. That's really simple because University of Bristol already has all of the legal agreements. So basically, you can just get the university to sign you up. You have to put in a project description in lay terms, which we put past an access review committee. Sensible projects all get approved. And if you ever need any help with doing this, that's the URL, the email address. You can look at current projects that are ongoing through this URL. So all of the projects that are ongoing are public. So I hope that I've encouraged a few of you who do common disease research, not just rare disease research, that you might consider using the URL. Thank you.
We got tough questions. Yeah, some people online, if they put questions in the chat, I'll look at them and ask them. Questions?
Yeah, a lot of people are interested in applying AI to these kind of data sets and trying to understand like variant gene interactions. Are you doing that in-house to try and sort of maximise the data you get, or are you looking for external people to come in and?
Look, our model is predominantly to use externals for all of the sorts of things that we do. We do have an internal AI program, but actually mostly we're doing this through collaborative efforts. And in fact, one of the research networks is specifically focusing on that. We also have commercial partners who are working on it. So, for example, we have a partnership with InstaDeep, which is looking at specifically at that particular issue. So we don't think that basically we should be doing these things ourselves, because the community outside is full of really bright people that we will never be able to match. And instead, what we should be doing is enabling people outside to be doing it. So we're absolutely up for that sort of thing. AI is an area that we're struggling a little bit with at the moment is machine learning approaches, where people build models and then want to export the models from our research environment, because we're very careful about ensuring that our data is completely de-identified and not identifiable. And deconvoluting the models offers a potential risk that people might be able to become re-identified. So that's something we've got to work on yet. But there's lots of other AI applications that you can do work within the RE, and ways, even if you are using machine learning based model building approaches, that we can ensure that your data goes out without it being running into our identifiability issues.
What's the one question? With the cancers you mentioned that have tumour sequencing as well. With the other data, such as the map, the whole methylome data, is that all blood, or are you doing other tissues as well as blood?
Yeah, so it's all blood. So the cancer samples are one of the negatives in the design of the 100,000 genomes was that although we collected additional samples on the rare disease patients, in the cancer patients, we didn't collect the tumours.
Okay.
And so we have DNA we had DNA that was sequenced. We don't have biobank of that. And so for us to go back would now mean that we'd be working off FFPE samples. ONT does not work with FFPE, neither does PacBio. It's highly fragmented, and the formalin itself actually bugs up the non-read sequencing. So it's not something we can return to. We have sought funding for a big multi-omics cancer program so that we can get long-read and other types of data from cancer samples. At this point, that's not funded by the government, but we're pretty confident we're going to be able to get a commercial program to do that. And if that's the case, we're looking at getting 8 cancer types, 500 cancers per cancer type, and then doing long read, bulk transcriptome, spatial transcriptome, proteome, liquid biopsy on them, and create a big multi-omics cancer data set. Of course, at the moment, although there's the Cancer Genome Atlas, it's actually using technologies which are many years old, bulk transcriptomes and things like that.
Yeah.
And there's a need for another multi-omics data set for cancer.
But what about sort of non-cancer diseases where, you know, the blood methylome is probably not relevant for the large majority of diseases here.
Yeah, again, it's what samples you've actually got. So one of the slides I skipped through is talking about a component of our generation study, which we've completely ripped off from LSPAC. It's called Babies in Focus. And
Sounds familiar.
imitation.
Very soon.
Imitation of
Blink of an eye and there'll be 30-year-olds in focus. That'll be a ghost quickly.
Anyway, this is a study where we're taking 15,000 of the babies and then using what samples we can obtain, basically profiling them very deeply. So the samples we know we're going to be able to obtain are cord blood and the Hill dry blood spot. We're hoping that we can also obtain stool samples, maybe placental samples, maybe salivary samples. So we'll see how we go with that. But that's the sort of thing that we'd like to get to.
John?
Yeah,
I'll just check on that.
a couple of things. Firstly, I'm right in saying that you're using your rare, in terms of using to look at common diseases, you're using people with rare diseases to also look at the common diseases in them. So I guess the question is, how you collected information about, because I thought when you refer people for genotyping, it seems to be, you know, they ask information about that specific disease, but what other, so you're collecting information about other diseases, and how are you doing that?
So we're doing it through data linkage. There's, I should be very aware.
Well, your friend.
I'm glad that it's not my friend. That's the last thing. It's just through the extensive data linkage capacity that the UK has built up through HDR UK. And so we have access to lots of hospital record and other public data sets, and shortly expect to have GP level data on these patients as well.
And I suppose one complication is if you've got a rare disease, is that right to affect your risk of other common diseases? Like, you know, if you're sitting around a lot because you can't walk, then you might have obesity and things. But I suppose that's up to people to have to figure out. But I guess, you know, how good is this collection of people with rare diseases as a way of looking at common?
Well, I guess that's why showing the idiopathic pulmonary fibrosis GWAS that I showed you, that it does actually pull up things that are relevant to common diseases. So there are always going to be issues in big biobank cohort studies with data cleanliness. That is absolutely the case with UK Biobank. And it will undoubtedly also be the case with National Genetics Research Library. And I guess that's the question, is to what extent you spend time trying to clean your data set and really make it tidy versus just the value of sample size. You know, there's that great parallel study when I was in Australia where Blue Mountain's Eye study spent more than a decade looking at orbital measurements, and then did a GWAS of orbital measurements and myopia, and published it back to back with the 23andMe study, which used the phenotype, do you use glasses to read, and found exactly the same things.
Yeah.
So, you know, I mean, basically, I'm in favour of bigger is better, and that actually trumps ultra clean most days.
Yeah. Just one other sort of practical thing. I mean, now it was a bit frustrating you referring patients because you had to go through a geneticist, and which I think the availability of genetic service varies, and how easily it is to kind of refer patients via that is, and then also, you know, you might feel sometimes as a specialist you know more about it than the geneticist. That's a barrier between the referrals. So that was a bit of a frustration. I don't know if that's in terms of trying to widen the access of genetic testing in the clinic. I think that would be useful to remove that barrier, maybe.
Yeah, so look, there are issues about how the GMS actually allows access to this testing, and then also how they allow us to give access to academics who are not the patient's clinician to identifiable data. And the patients consented in the first place for a research study which said that their data would not would become identified. And so we're now in a bit of a bind because many of them would actually like their data to be identified, but never consented that that would be the case. And so what we do is we go back and get individual consent when that's the case. So we have a team of people who work so that when researchers want to identify a patient, that we go back to the patient and ask them to consent for that data to be made available. It's extremely laborious and it's not ideal, but it's historic because of the way the consenting was done. In terms of mainstreaming, which was sort of where you were getting at a bit as well, undoubtedly this is going to have to move away from this being something which is where the gatekeepers are clinical geneticists. So the 150,000 people who are being done for the Adult Population Genomics Project are going to be recruited from the general community and from general practices. And the feedback of their genetic data is going to be in simple ways, online and through general practitioners, not through clinical geneticists. And that sort of thing has to happen. Now, if we get to the point where half of all healthcare episodes by 2035 involve a genetic test, there's no way that scales if it's all done through clinical genetics.
But I think there's a sort of middle ground, not just sort of in GP land, but when you're in a specialty clinic and you're seeing quite rare, unusual or severe cases and you're also managing secondary care, you know, it's quite helpful to I think we need to be facilitating the genetic analyses and referrals in that set. And I think that having to say you have to refer to geneticists first is currently a barrier.
Just to briefly, just because I lived in Bristol, worked in the genetic service here for about 7 years before I moved to Genomics England, and was leading the team that helped to recruit to the 100,000 Genomes Project in the west of England. That was some years ago now. We finished recruitment in about 2017-18. But I'm sort of searching back in my brain here, but I would say we probably had at least sort of 40% of those referrals for rare disease patients that came not directly via clinical genetics and came via other like rare disease specialists. Many of them had had some involvement with clinical genetics before as well, or they were part of that sort of broader referral. But the 100,000 Genomes Project was where at least the NHS England was first starting to make that push to say, as you're saying, clinical genetics can't necessarily be a referral barrier because we need to start educating other clinicians to recognize those conditions if they don't already. And because the waiting list can be really prohibitive then to patients getting an answer. So 100,000 Genomes Project kind of started to do that. And hopefully, I don't know what it's like as much now in the Bristol area, that for patients being offered whole genome sequencing for rare disease, that's increasingly coming through different specialists. But it really varies also depending on the region, how the genetic services are set up in that region, and how specialists sort of feel equipped to do the testing directly themselves.
So a question online from Nick Timpson, who says, brilliant presentation. But one question is, will the adult population study be volunteers? The adult population you discussed at the end.
Well, I'm just wondering what the alternative is, Nick. What are we going to do? I don't think we can conscript for this sort of study. They're definitely going to be volunteers. So I'm not sure where that's coming from. But
I mean, you can approach people and then they have to consent, but you actually select a group of people who you ask, or whether it's or whether it's volunteers who just come forward, like in the recent chronic fatigue study, for example.
Okay. I don't think we're just going to be writing out there and just our future health style, just taking all comers. Instead, basically, we'll be going to targeted communities associated with particular general practices, for example. So general practice lists to try and recruit patients through them. Because what we're really interested in this study is to see whether, how GPs use the data and what impact it actually has on health outcomes in relation to GP prescribing. So it won't be strictly a population level study.
No,
It will be weighted towards ill health.
but through general practice.
Yeah.
Yeah. And then the next other question was, regeneration study. Could you comment on what will happen to this cohort, re-affiliated data, linkage data, and beyond?
Yeah, I can comment on that. So we, the anticipation is that that cohort joins the NGRL, the same as the other cohorts, so the same linkage to the existing data sets we have would apply. We have looked to introduce additional data sets that we know would be helpful for that linkage, like the maternity services, community services data set, and also exploring whether there are additional data sets beyond that as well that would be useful for this cohort. But that's also part of the discussion that I think will be ongoing through Babies in Focus. So the initial, when the initial participants' data is in the research environment, it'll include the existing data sets we have, but then there's the potential to build that over time.
Hi, so we seem to have moved on a little bit to data linkage, which is what I do with children of the 90s. Just wondering if you've looked at data linkage sets beyond health, for example, education, so that you can see the long-term action impacts perhaps of these conditions.
So at this point, we've not linked it up to educational outcome. That's a very sensitive area for our participants who are anxious that basically that the genetics doesn't get used, for example, to stratify people according to educational potential. So I think it's not an impossible thing to do in future, but at this point, it's not a high priority thing and one that would take a lot of engagement before we were to do that.
Any other questions?
That's a good practical question about, it sounds like a good idea combining NGRL with the UK Biobank data, but they're both in protected research environments, and how practically you'd be able to do that, like, apart from using kind of summary level data.
Yeah, you have to use summary level data. Working out a way that people can actually work at individual level data between the two is a high priority for the big three, Future Health, Genomics England, and UK Biobank. There are plenty of others as well, so I'm not saying we're the only group, but the three cohorts have been looking very closely to see how we could do that. We would love to be able to do that without putting it in the one TRE. But you'll be aware that GA4GH has got different approaches to potentially allow people to shift data between one and the other. And so we're optimistic that we will be able to do it. At the moment, it's just summary level data.
There's no other questions online. So we thank Matt again for a really great talk.